Good morning to everyone. Today we have discussed on the process development of the heterocyclic drug containing pyrazole and piperazine framework. Outline of the proposed research work contains first problem, the high cost treatment during the early phase of the introduction of medicine by the innovators, increasing demand for lower cost of generic drugs product. The most important challenges of the process development includes patent infringement issue with respect to process, intermediates, polymer, particle size, reagents, impurities, and solvents, growing regulatory concerns, control of genotoxic impurities, polymorph issue, developing efficient HPLC-GC method for analyzing the intermediates and APIs, green chemistry challenges. The literature survey for the synthesis of biologically active heterocyclic compounds containing pyrazole and piperazine frameworks EPICS is performed, and based on the literature survey following is observed, reported process has low yield and makes use of a costly raw materials. Reported processes makes use of a column chromatography for the purification of intermediates and active pharmaceuticals intermediates. Reported process does not provide any information on the formation and control of the impurities. No description about the polymer obtained by the following the innovator process is available in the literature. Few of the processes are patent protected and hence cannot be opted for the synthesis, and thus it is essential to develop the non-infringing process for active pharmaceuticals APIs. Reported synthetic scheme as per the literature for the synthesis of first biologically active heterocyclic compounds containing the pyrazole framework is apixaban. The disadvantage of the reported synthetic scheme is in a red color. The overall yield was low. Use of a carcinogenic solvents is chloroform. Use of a hazardous raw material. Use of a column chromatography technique for the purification. No description of impurities and their controls is reported. No analytical methods are reported. Reported synthetic scheme as per the literature for the synthesis of the second biologically active heterocyclic compounds carry Piperazine hydrochlorides. The disadvantage of the reported synthetic scheme, overall yield for the two disclosed methods is very low. Use of expensive catalyst, example BNAP. Use of a column chromatography technique. No description of the impurities and their biologically and their control in the biologically active heterocyclic compounds. The main objective of the research are as below: development of the non-infringing process for a few biologically active heterocyclic compounds with minimal cost, establish the process optimization strategy to achieve the higher yield and controlled related substance for the biologically active heterocyclic compounds, identification, synthesis, and characterization of the impurities of biologically active heterocyclic compounds to achieve the desired quality of biological active heterocyclic compounds as per the pharmaceuticals guideline meets and also meets their regulatory norms. We have hypothesis study. We have first biological active heterocyclic compound developed. We have proposed synthetic scheme for the synthesis of first biological active heterocyclic compounds for apixaban, in which we have first the morpholization. After the morpholization, demorpholization of the material, demorpholization, then the reduction of the nitro group. Nitroamination undergoes the N-acylation reaction. N-acylation reaction followed by the cyclization provides the apixaban, which is a biologically active heterocyclic compounds. We have also proposed the synthetic scheme for the synthesis of secondary biologically active heterocyclic compounds carry Piperazine hydrochlorides. The methodology through the literature search will be done to identify the infringing issue for the synthesis of biologically active heterocyclic compounds. Based on the literature search, key starting material will be identified for the synthesis of biologically active heterocyclic compounds. The process optimization plan will be designed for the synthesis of intermediates and biologically active heterocyclic compounds. The synthesized intermediates and biologically active heterocyclic compounds will be analyzed as per the valid test method for the impurity profile, assay, and other analytical test as per the requirement for the active pharmaceutical ingredients regulatory norms. Tentative chapter involves the chapter 1 case study for the process development of the biological active heterocyclic compounds containing a pyrazole framework, that is apixaban. Case study for the process development of a second biological active heterocyclic compound containing the piperazine, cariprazine hydrochloride, and its publication and patents. Thank you.